Hello and welcome to episode 15 of The Future of Drug Discovery. In this episode, we speak with Björn Schimmöller, the co-founder and CEO of iuvantium, a synthetic immunobiology company developing new modalities for the next generation of immunotherapies. Bjorn is a process engineer and material science expert, having stemmed from working in a world-class inorganic chemistry laboratory, the Sotiris Pratsinis Group, at the Swiss Federal Institute of Technology in Zurich. In addition, he has years of commercial experience leading and developing new nanomaterial technologies with companies including the Cabot Corporation. iuvantium is gearing up for some big changes in the sector in the coming years, and this is undoubtedly an episode you won't want to miss. Enjoy.
Thank you for having me on the podcast. It's really great what you do, and I'm happy to talk a little bit more about iuvantium and what we are about. So iuvantium comes from the Latin word iuvare, which means to help or assist. When we started working on our technology, one of the obvious applications is in vaccines and adjuvants that help the antigen work. And because our platform is not necessarily limited to vaccines, their broad application space as a platform technology, we didn't want to be confined. So we decided what we do is we help patients. So iuvantium seemed to be right. And if you put iuvantium mundum out there and stay all in Latin, it means helping the world. And that's our mission. We want to help as many patients as possible to get cured so that they can live a fulfilled life and raise to whatever their potential is.
Amazing. What would you say were the key events that led you to this moment?
That's a good question. There are lots of things that had to happen in this order in my life to get to the point where we are now. Serendipity is, to me, one of the most powerful things in life and the professional career. If you could engineer serendipity, if you can increase your chances for something to happen, you're doing really well. And in my case, I studied in Germany. I moved to Switzerland to do my PhD. I took a job in 2010 in the US, where a lot of my friends told me I'm crazy to go to the US with employee protection and all of these things. But then I came to the UK because I didn't feel fulfilled anymore in the job that I was doing. I wasn't really aligned with the company strategic direction and how they were doing business. I looked for another job and I ended up in Oxford, of all places. And it happens to be that Oxford, especially during the pandemic, was a very center point of what was happening in vaccines. And my company at the time became the leading manufacturer of the AstraZeneca vaccine in Southeast Asia. And because of that, they were suddenly interested in what can we do in material science and life science. This technology came on my desk that was developed at Oxford University by my now co-founder, John Austin, who was professor for immunobiology, and by Professor Dermot O'Hare from the Inorganic Chemistry Department. And they have worked on that technology in 2009 for the first time. And from my perspective, not knowing anything about biology, life science, immunology, that was not my world. My world was functional materials that go into plastics, LCD screens, or laser printers or so. But I saw a material that had very intriguing properties, very intriguing functionality, and that was an inorganic material that was used to stimulate the immune system. At this point, when I realized that I understand the material side of things, John understands the immunology of it. If we combine a chemical engineer, material scientist, and an immunologist, and we develop something that is unique and is not really pursued in the industry, something special may come out of it. We did a project, and the next serendipitous event was that 2022 was a tough time for the chemical industry. My employer went through a hard stretch. They were saving costs on all corners and decided to abandon what was their most ambitious and most non-core technology adventure. And they wanted to focus, understandably so, on their core business around sustainability, circular economy, because they were making primarily polymers. They have a lot of challenges around CO2 and energy savings, etc. So that was not what I was interested in. And I said to John, if we believe in the technology, you from the immunology side, I'm from the materials perspective, the only way to commercialize this and to get it to the patients is we found our own company. And you have to consider John was looking back on now a 40-year career at Oxford University. He was one of the first postdocs of the late Ralph Steinman, the Nobel laureate. He was ready to retire and enjoy his life. So he said, okay, if you give up your job, I will postpone my retirement. Let's do this. So here we are. We are getting close to a year since we incorporated. We have learned a lot and we have made a lot of progress. We have recently welcomed a new team member, Farouk Khan, who is the guru on artificial intelligence and complex systems, who has really helped us to shape the idea for where this company and this technology can go. And we are super excited about all the different things that we can do, but we need to be able to prioritize and focus as well. I think we have a lot of promise ahead of us.
They say a CEO has 3 areas of accountability: vision, building a good team that can deliver on that vision. It seems like you have the core team in place. And the third aspect is the funding to get the team do what they need to do to achieve that vision. How are you guys doing on that front?
As first-time founders, we're learning a lot. What we didn't know when we started in December, that we're going to hit the worst funding environment in a long time. And we're learning by having a lot of conversations. If I reflect on some of the things, we started way too early to talk to investors where we haven't had yet clearly defined who we are and what we can do and how we want to do it. But it's established relationships and it allowed us to iterate. In the early phases, you iterate a lot. And it is important to get feedback. What are the investors interested? What is fundable? What is not fundable, especially in this environment? And we have now gotten to a point where I'd say we have a clear definition of who we are and what we want to achieve. We have a clear path of what are our next steps. And the conversations that we are having now are much more meaningful, but we're still looking for that lead investor who is buying into the vision, buying into an approach to vaccines and therapeutics that no one really has looked at from an inorganic chemistry perspective. Everybody is working on organic chemistry. We're saying that the history of vaccinology and immunotherapies was written by antigen and antibody discovery. Everybody is trying to find the next antigen, the next antibody, the next ADC or CAR T or so. We believe that your immune system is the best chemical factory that you can have in order to generate those. We want to stimulate or program your immune system with what we believe a very unique materials platform using inorganic chemistry. And getting across that story, getting across to investors who are following a very rigid script in terms of what a therapeutic company should be, or in your case, what tech bio company should be. If you come out from a left field, it's always challenging to find the right way to communicate that. Your history in terms of being an electrical engineer and moving into bioinformatician is interesting because we are a motley crew of people coming together that have merged into a very precise thinking of what we think we can deliver. Our selling point is we believe that the future of drug discovery is not only focusing on the active part of an antigen, an antibody, a checkpoint inhibitor, but is to figure out how you can augment the immune system to help you bring it to an efficacious state. And we believe that inorganic chemistry can play a very important role there because we have a technology where we can control quality and quantity of immune responses purely based on physicochemical properties. And we can predict the immune response our materials will elicit in a very straightforward way. In the chemical industry, you are used to the first law of thermodynamics. You are accustomed to Navier-Stokes equation and fluid dynamics to predict almost anything, mathematically describe a chemical reaction or a flow field. Entering now biology, where I would say the common opinion is biology is too complex to be predicted, is fascinating because I have a very different mindset. If there is a way, if you could mathematically describe something, you can predict it. And we think we have found something there where we can mathematically describe the immune system. And we're building upon that to really make a change, enable the next generation of vaccines and immune therapies.
Have you narrowed down your focus for the first application of this technology?
That's one of the things that teams in general that want to build a platform struggle with. You see all these opportunities, but what makes sense to start first? We decided to focus on vaccines, mainly for 2 reasons. One is vaccines is well respected and understood that an adjuvant can help the efficacy of a vaccine. So if you think about your standard flu vaccine or an HPV vaccine or a strep vaccine, they all have an organic compound in it, which is the antigen that targets the pathogen, but they also have an adjuvant that is basically the clumsy burglar that is breaking into the house and raising the alarm. You need that in order for the antigen to be picked up by the immune system. So the notion of adjuvanting an antigen in vaccines is common knowledge. 70% of all vaccines are adjuvanted. A large fraction of those is adjuvanted with an inorganic material, which is aluminum hydroxyphosphate or aluminum hydroxide. So it's also already an inorganic material. And then the other thing is time to market. So you cannot get regulatory approval for a standalone compound. It always has to be in the formulation with an antigen, a new vaccine or a new immunotherapy. And that would mean the development timeline is somewhere between 7 and 12 years. But vaccines have an advantage there that if you have already a vaccine on the market and you want to establish a new version of it, you don't have to go through the whole clinical trials. So you can get with a minimal clinical trial where we only have to show that it's not worse than what you already have on the market, you can get approval. So we think that companies that have vaccines on the market or potentially already in clinical trials could be interested to add our technology, augment their formulation, and then you can get into market a lot sooner. And there are plenty of examples of vaccines that facing a patent cliff in the next few years. And these are blockbuster type vaccines. And the companies that are in charge of those, if they find a way to extend patent protection while keeping all the supply chain and quality control, etc., the same, I think there's potential for a drop-in solution. How we call it in the chemical industry, something that is already fitting an existing formulation. That's our strategy. And once we have shown the applicability and the capability of the technology in terms of steering and polarizing immune responses into a certain direction, the rest of the industry will realize what could be done with that if you combine it with other therapeutic approaches.
You said that the new clumsy burglar you designed shouldn't be worse than what they already have, and hopefully it will be better. And when you think of a betterment, I'm thinking you can create the same efficacy or a higher efficacy with the same dosage or lower dosage, maybe reduce toxicity, higher attrition rates in clinical trials, and that would be significant. Is that something that you're planning to test? And if so, it's going to take a lot of time to prove that it works. So how are you going to tie that outcome to the development of the company?
That's a good question, one that is not so easy to answer. You're touching on what the long-term vision for the company is. I firmly believe that we have to go beyond just a discovery phase. We have to make an impact and change the probability in clinical trials. So what do you have to do as a company? Where do you have to play? And if you think about the whole process development or drug development pipeline, at which points would you create the most value? I think it is in the clinical trial stage. And so we want to develop technology where we can reduce uncertainty in the clinical stage. One way to do that is being able to predict the immune response that our materials will elicit, and then use that with assets that have failed in clinical trials because of toxicity or because of efficacy. So the company can have a perfectly fine antigen that is the perfect target, but they need to dose it so high that you get adverse effects. If you can augment that antigen with a way to boost immune response but lower the dose, you have potentially resurrected an asset. On the efficacy side, it's confirmed that the antigen is working and harmless and non-toxic, but you're not getting the right response on the immune system. So you have to help that. And if you get a non-toxic material to do that, you can resurrect an asset that failed because of efficacy. By doing that, you have bypassed years and years of preclinical work already because the targets are known. You just need to find the right partner, the right burglar to raise the alarm.
So you have the solution to create new materials that can help to elicit higher efficacy. On the other hand, you have a lot of expertise in how the immune system operates.
Yeah.
Are you saying that you would be able to predict and test the outcome of the new materials on the immune system without even going to clinical trials?
Yeah.
Okay. Would you be able to tell more on how that works and how long does it take to do a single cycle of predict the new material, test if it improves the efficacy or not, and feedback into the system?
The timeline is not that long. What we are able to do is synthesize a new material, We have complete control over the chemical composition, over its size, over the physical properties of it. And based on knowing those, we can already predict what type of cytokine response it would elicit in a human dendritic cell and in a mouse. We have done that proof of concept already. So where we want to get to is we need to feed our algorithm more data to then be able to reverse engineer it. We want to get to a point where we know which output we need from an immune system. Do you need an IL-6? Do you need an IL-12 or 17? Or what type of antibody? And then be able to generate the material that would deliver that type of immune profile. In terms of the cycle, we can do what I just described in synthesizing, predicting, and measuring in a few days. Getting to the point where you can say, I want this profile, I want this quantity and quality of an immune response. Tell me what type of material we need to synthesize. We're not quite there yet, but we hope that we get to that point sometime next year. What we believe we have found is what we call a biophysical law. John has seen 40 years of dendritic cell therapy development. He has worked in immunology for so long. This was the first time somebody was able to predict what the immune system is going to do. This is where we see so much opportunity to take biophysical law and apply it. For our compounds, we are more or less watertight. That we can do that right now. But how do you start understanding the immune system, decoding it, and rebuilding it as a digital twin? To me, the foundation is to have a mathematical description of it, which we have now. And it will allow us to start putting the puzzle pieces together. There are other people like yourself working on the antibodies. There are companies that are putting other puzzle pieces together on a completely different corner. We may not even see each other because it's so complex. But if we start building at one corner, eventually all these pieces will come together. And that's our vision, is to decode the immune system and be able to build a digital twin. And then you can start putting the omics into play, the epigenetic data, the genomic data, and we're building a digital twin of your immune system. And then we can go out and say, what is the best therapy available or to be designed therapy to address Björn's disease that he needs to fight? How do we program his immune system to do the job? We have a mathematical formula. That's like the foundation. That's all we need. And so you have John, who is still grappling with the idea of, can we actually do that? Could we actually get to what we're describing? And to me, that's the power of what iuvantium is doing and the power of the team. We're bringing 3 people together that are thinking very complementary about what may be possible. And we are not afraid to ask these questions and say, yeah, why not? What is there that could go wrong or prevent us from achieving this? And so far, we haven't found anybody who is able to pull the cart on the bottom and let the whole house of cards crumble. And I hope we're never going to find that person. But yeah, that's part of the excitement, I think.
It really captures my imagination hearing companies or ideas like yours, which maybe falls under the category of immune system 2.0. There was one many years ago from this institute. They were planning to develop a hydrogel that you put into the human body, which then attracts your T cells. It further programs them based on the antigen that they were not able to detect before, and then release them when they reach the right amount of population and go and kill it. So all of this stuff is so fascinating. And I think generating with the hype terms like foundational model for the immune system, the value of it is incomprehensible. It's a journey that we will be closely watching, and we wish you all the best for that. Is there any call to action from your side? You're probably building a bigger team, more funding, partnerships. If there's anything else that our audience can help with, what would that be?
So if there's an investor listening, we are raising a seed round at the moment and looking for a partner that shares the vision and the mission and is not afraid of taking on a different approach to drug discovery. And then in terms of the community, what you're doing is bringing people that work on different areas of the same problem, bringing them together and stimulating discussion and challenging each other with some contrarian thoughts or sharing what new technology is out there. So I'm welcoming anybody who is interested in what we're doing or who wants to engage and partner with us. As the CEO, I have to be a little bit paranoid. I'm still waiting for that person that comes across and tells us your technology is not going to work and here are the reasons. So, and obviously, the sooner we find that person, the better. Or hopefully we never find it. We want to grow and we want to build a company that is in it for the long game. We're playing I don't know if you are aware of Simon Sinek. He's a very big proponent of the infinite game. And I think that's really where we want to be. We are not going to be getting where we want to go on our own. We need partners, we need collaborators, we need a community that is challenging us and itself to come up with new ideas and new ways of approaching things. So anybody who wants to reach out is more than welcome. I'm first-time founder and still learning a lot. And there's nothing better than to learn from people that have gone through it, that have experience, and are willing to share those. As a community in the drug discovery and life science, we will make great strides in the future.
Yeah, I believe in that 100%. Bjorn, it was a true pleasure having you. I learned a lot. I'm sure the audience will too. Thank you very much.
Thank you, Murat. I really appreciate it. And it was great fun. I would love to do it again in 2 years from now. We'll see where we are and where you are. We're going to follow Antiverse very closely, and good luck with everything in Boston.
Thank you. Very kind.
Thanks for listening to this episode of The Future of Drug Discovery, brought to you by Murat, the co-founder and CEO at Antiverse, and the production team here at Antiverse. We value your support immensely, and to help us continue growing this vibrant community, we kindly ask you to like, rate, and review the podcast. Your feedback fuels our mission for a better connected community. Also, before you sign off, did you know that every month we host a free live event with an industry leader and/or expert in the drug discovery space? It's the perfect opportunity to meet other community members and to build your network. We even host a 90-minute virtual networking session to help bridge innovation in the community. To be notified about our future events, follow us on LinkedIn and Twitter, or opt in for email updates using the form in the episode show notes. We hope to see you at our next event. Until next time.